Hi everybody, welcome to the final live stream of fall quarter. Crazy, went by so fast for me. That's awesome that we're here. Today we're going to be talking about gene expression. Gene expression, in a nutshell, describes why we turn on some genes and why most of the time genes are not turned on. We don't need all of our genes expressed all the time. It's a waste of energy, and it's really just not necessary for our system. So gene expression is really great about keeping things on when we need it on all the time, and keeping things off when we don't need it on all the time. So generally, an analogy I use is, when I'm doing homework in my room at night, I'm going to need my light on, but if I'm leaving my house, I can turn it off, so I don't need to express that light all the time. So let's get started. We start with a chart to talk about the lac and operons, lactose and tryptophan operons. And remember that operons are a cluster of functionally related genes that can be controlled by a single on and off switch. I wrote a couple of things up here to get us started in looking at the difference. And it just starts in the table, so I'm going to start from the first part. Is the metabolic pathway anabolic or catabolic? In lactose operon, it is a catabolic pathway. It hydrolyzes or breaks down lactose, a disaccharide, into glucose and galactose, 2 6-carbon sugars. We're going from something bigger, which is this disaccharide, and we're breaking this bond into two things, so it is a catabolic reaction. Another neat thing to know is that when there's an increased concentration of lactose, or lactose is present, generally, with a few exceptions that we'll talk about when we get into it, the operon is on. Whereas for tryptophan, it is an anabolic reaction, so it synthesizes tryptophan from precursors. Tryptophan is very necessary in a lot of processes that go on in the body, so it needs to be produced all the time. It's a lot like, it's not similar in function to glucose, but we need glucose all the time. It's used in a lot of things. Tryptophan is similar, and that's needed. So when there's a decrease in tryptophan concentration, that's when the operon is on. Okay, so what are regulatory genes associated with the operon, and what functions does each serve? The genes associated with the lac operon is the lac-I promoter, CRP binding site operator, and these are the genes. The functions of this operon is that the lac-I produces an active repressor protein that binds to the operator in the absence of lactose. And the promoter is the site where RNA polymerase binds to the DNA, and binding of RNA polymerase is enhanced when CAMP interacts with CRP and forms a complex. binding. And just a little reminder down here, CRP and the complex bind at the CRP site on the promoter region, and as a result, the enzymes for lactose digestion are produced only when needed and only when glucose is not present. So they need, the CRP and complex need to bind on the site of the promoter region, and so that's why they're only produced at a certain time, because the concentration of glucose also directly relates to the concentration of cyclic AMP, and that's how that is related. Alright, moving on to tryptophan, and hello everybody, thank you for coming. The genes associated are tryptophan are promoter and operator. And their functions are that trypt-R produces an inactive repressor, and this binds to the operator only when there's excess tryptophan. So this binds to turn it off only when there is enough tryptophan, there's enough out there for it to use. And these little boxes around here, it's just, that's just to say concentration of tryptophan, is what that means. RNA polymerase binds at the promoter site and transcribes the genes for making tryptophan as long as there's a low concentration of tryptophan, or as long as tryptophan is not in excess. Okay, and then just sliding this down a little bit. What structural genes are included in each operon and what does each produce? And we see here that the genes associated with lactose are LAC-Z, Y, and A, and the products that come from those are that LAC-Z codes for the B-galactosidase enzyme, which digests lactose into glucose and galactose. LAC-Y codes for permease to allow lactose to enter the cell, and LAC-A codes for transacetylase, and its function is unclear or unknown. For tryptophan, the genes are Tri-A, B, C, D, and E. And the products for these 5 genes are that they code for 5 enzymes required to convert a precursor molecule to tryptophan. And just a reminder, tryptophan is an amino acid required for protein synthesis. Awesome. And then, is the operon inducible or repressible? The lac operon is inducible. And just remember, an inducible operon is when a repressor is bound, and then an inducer binds to the repressor, inactivates the repressor, and then genes can be expressed. So let's say that we have genes here. These little lines mean that all of these are the different genes. This is our operator, and we have this repressor bound. Basically what happens is that something comes in here and binds to the repressor, changing its shape so that now the operator is free and genes can be expressed. Oh, I'm now seeing that you guys can't really see that. Sorry about that. Anyway, I hope that picture is clear. So this little thing, this thing right here, is an inducer. Awesome. And then for the trip operon, that this is repressible, which you can think about the opposite. So this is always off because the repressor is there unless the repressor is taken off. With repressible, it's actually always open, and then a repressor has to come in on the operating site to make it so that genes are not expressed. So it's usually in the on state. I'm going to put expressed here, expressed here. Oh, after tryptophan, it says AA, required for protein synthesis. That means just it's an amino acid. Tryptophan is an amino acid required for protein synthesis, is what that says. And so to clarify this, an inducible operon is usually off because it is repressed until an inducer comes in, removes the repressor, and then genes can become expressed. Genes can be expressed, sorry. A repressible operon is one that is usually expressed, but a repressor can come in and make it so the genes are not expressed. Does that clarify those operons? Yeah, of course, and I can also stop being lazy and just write out amino acid. I do that in my notes all the time. I'm taking biochem, so we say amino acid all the time. Okay, awesome. I'm glad that clarifies. The next question is the repressor protein produced in active or inactive form? And remember, this is lac, this is tryptophan, that's a Y. The repressor protein for the lac operon is produced in its active form. Remember that the repressor protein binds to operator to stop transcription of the genes. I don't know why I did it like this. I didn't have a question. Okay, and then for tryptophan, the repressor protein is produced in its inactive form. Number 1. Under what circumstances would the lac operon be on versus off? And the same question for the trip operon. For the lac operon, it would be off if there was no lactose in the cell. And it would also be off or operating at very low levels, even when lactose is present, if there is sufficient glucose present as well. And this just means levels. And then it would be on when lactose is present and there's little to no glucose. As you can see here with the off and on, they kind of go with each other. So no lactose, sufficient glucose, some lactose, sufficient glucose, it's off. And then if lactose is present and there's little to no glucose, then that's the only time that it's on. The trip operon, and I don't know why I keep writing try. It's because trip is actually W, and it's always, I always like mix them in my brain, goes to Y. I don't know why. Anyway, the trip operon would be off when tryptophan is readily available. It is on at pretty much all other times. Awesome. How are the lac and trip operons similar in structure, function, or both? Both have regulatory genes. That produce repressor proteins that can interact with the operator. And stop transcription. And both of them have a repressor. The repressor just works different ways in both. Okay, number 3. What are the key differences between the lac and trip operons? The lac operon, I'm just going to do it again. Put them on two different sides so we can compare. We know that the lac operon is inducible, so the presence of lactose induces production of the enzyme needed for lactose digestion, whereas the tryptophan operon is repressible, in which it's ordinarily on, producing tryptophan, which is needed for protein production by the cells, but it's turned off or repressed when there's an excess of tryptophan available. The lac operon is controlled by both a regulatory protein that acts with the operator and blocks RNA polymerase action, and a CRP site, so that RNA polymerase does not match with the operator unless CRP in its complex with CMP is attached to this site. So, to write that down and make it a bit easier to digest, it's controlled by a regulator protein. That binds to the operator and blocks our RNA polymerase and a CRP site. I like pictures, so I hope that this is helpful. But if this is the operator and this is CRP, and just remember that CRP enhances the interaction of RNA polymerase with the promoter region, and so this is really responsible for just making sure that that connection stays strong and keeps going, and it really pushes for the expression of those genes to happen. And so when this repressor protein, which I'll call RP, binds to the operator and CRP, then the RNA polymerase here can't bind with the operator anymore, and that's what controls this protein. Another note here is that CMP levels are low when glucose is present, and CRP and cyclic AMP are what forms a complex at that site, and that's what enhances the expression of lac operon. And this makes sense. So if glucose is low and cyclic AMP is high, then it's going to encourage a cell to express the genes of the lactose operon. It's going to break down lactose into glucose and galactose. So now we have glucose again. We had a low concentration. We're trying to replenish that concentration. And that's the more confusing one. Honestly, tryptophan is repressible. It's only turned off with an excess of tryptophan. Alright, next question is, what advantages are gained by having genes organized into operons? And just to reiterate, operons are a cluster of functionally related genes that can be controlled by an on-off switch. And so some advantages is that as they're needed, both systems are set up to simultaneously be on or off all of the genes that are required in a metabolic pathway. So this is more efficient than having a gene under independent control where you'd have to go through every individual one and turn this on and then turn this on and then turn this on. It was like how you see those old movies of Frankenstein and he's turning all of the switches on his lab, and instead of that it was just one light switch. That's kind of what's going on here. So to write that down, both systems are set up to simultaneously turn on or off the genes in a metabolic pathway. Shoot. And this is for efficiency's sake. Okay. The next question. Strain X of E. coli contains a mutated lac regulatory gene on its bacterial genome. As a result, the gene produces a non-functional lac repressor protein. You add a plasmid, an extracircular piece of double-stranded DNA, to these cells. The plasmid contains a normal regulatory gene and a normal lac operon. Build a model or diagram of what one of these modified E. coli cells would look like. Then answer the question and use your model or diagram to explain your answers. So E. coli are little bacteria, and I can draw them just up here. They have circular chromosomes. So let's say this is bacteria 1 and 2. They have circular chromosomes on the inside. And let's say that this mark right here is in one where the defective lac operon is present. Maybe actually, let me choose a better color, but the picture is going to look exactly the same. So when this, when a plasmid inserts its DNA into this gene to make that lac operon functional, it's still going to have that other gene, but now it has the functional. So this gene right here that's functional is able to break down lactose, is able to repress the breakdown of lactose, and the non-functional is not, because remember, as said here, all that mutation does is create a non-functional lac repressor protein. So before the addition of the plasmid, would E. coli strain X cells be able to produce the enzymes for the lactose digestion? Explain. The answer to this is yes, because the lac gene ordinarily produces an active repressor that inhibits production of the genes for lactose digestion, but in this case, the gene is mutated, so it can't produce the repressor, and lactose digestion would be able to happen within the cell. Awesome. And I just dropped that form in the chat, just so you guys know. B, after the addition of the plasmid, so this fixes the repressor protein, would the plasmids lack operon produce the enzymes for lactose digestion constitutively all the time, is what that means. Or only when lactose was the available sugar sucrose. So the plasmid contains a normal regulatory gene, so it's a normal lac operon, and as a result of this, the plasmid's lac operon should produce genes for the lactose digestion only when lactose was the available sugar source. So not constitutively. Awesome. C. After the addition of the plasmid, so we have that working repressor, would the bacterial genome's lac operon produce the enzymes for lactose digestion constitutively or only when lactose is available energy source? So this is pretty much the same answer, because energy source and sugar source can be viewed similarly when we're talking about the need for glucose. So after the addition of the plasmid, lactose digestion happens only when lactose is the available energy source. And D. If equal amounts of lactose and glucose were present in the cell, would the lac operon in the bacterial DNA be on or off? Would the lac operon in the introduced plasmid be on or off? So in the original bacterial DNA with the mutated repressor, the lac operon would be on because the repressor is not able to be made. Is unable to be synthesized. And then in the introduced plasmid, remember we said that, sorry, I was just getting my sentence together. So we said that it would only be on when it was pretty much the only source. So when glucose and lactose are at equal levels and the repressor can be made, then the operon would be off. Because glucose means low cyclic AMP levels, which means no enhancing of RNA on the lac genes. Okay, so now we're going to move on to gene activity and its control in eukaryotes. Sorry, just getting this ready. And first, this says human genes cannot all be active at the same time. If they were, all the cells in our bodies would look the same and have the same functions. For specialization to occur, some genes or gene products must be active while others are turned off or inactive. Complete the table below. And just note that the table represents, it's a representative sample of the various types of control mechanisms, but it doesn't include all of the possible mechanisms of control, or we could write a novel about it. So starting with A, the GNA or DNA itself. So chromosomes or parts of chromosomes can be inactivated by DNA packaging as heterochromatin. We have urochromatin and heterochromatin. If we look at a chromosome, usually this is where urochromatin is, right by the centromere. And this is where heterochromatin is. And it's just inactivated DNA. So the urochromatin, like this bunches right here, are really what is used. And then another mode of control at the gene or DNA itself is that transcription from DNA can also be controlled by enhancing or inhibiting the action of various proteins that act with the DNA or RNA polymerase. So that can make it more or less likely to produce an mRNA transcript. Awesome. And then the mRNA product of the gene is the next site of control, so that can be controlled at the mRNA level. Controls affect whether pre-mRNA is processed into mRNA. So control is happened to. Pre-RNA is processed into mRNA, and how long the mRNA is active in translation. And whether the mRNA is capable of being attached to the ribosome and translated. Okay, and finally for C, the protein product of mRNA. The protein product can be made inactive form. So a protein can be made either where it's an active form or in its inactive form. So its inactive form becomes active only in specific environments. And an example of this is digestive enzymes. They're usually inactive when they're produced, but when they get to the pH that they like, which in the stomach is very low, it's an acidic environment, once they get into that environment is when they become active and can start working. If produced in active form, then the protein can be inactivated by the presence of a molecule that changes its allosteric form. And another thing is that it could have a limited lifespan. A self set to destruct button after it's done doing its thing, so it could have a limited time of functioning in the cell. Awesome. So moving on to the next question. And D cut off of this, so I just wrote it at the bottom here in red. Hope that's okay. Number 2. Sickle-celled organisms such as amoeba and paramecia often live in environments that change quickly. Which of the following types of control allow organisms like these to respond quickly to frequent short-term environmental changes, and explain your reasoning? So the answers to this can be just different depending on how we can talk about short-term. Short-term evolutionarily can be very different than generationally. Generation is just one to the next, and evolutionarily it can take a very long time for things to change. So in the short-term response, going through these, The control of enzyme concentration by controlling the rate of mRNA translation maintains a certain number of different enzymes. And it's energetically efficient only if the short-term changes occur frequently over time. So frequently over time is when it's efficient and when it can produce those short-term changes. Control of activity of existing enzymes is really great for responding to short-term environmental conditions. And so this could definitely be something that helps these little critters change so frequently. And I saved A and D for last just to talk about them a bit more, because the control of mRNA transcription from DNA, if you don't already have the machinery, so B and C are talking about mRNA already existing, or these enzymes made of proteins already existing. They're already out there and they can change really fast. But if you have to start from square one, from translating mRNA from DNA, it can take a while if it's a short time. But in general, this wouldn't take too long. It would just require a lot of energy to do so. And then D, the control of the amount of DNA present wouldn't really help quick, short environmental changes. You can't change your amount of DNA. However, in this question, if you only have a certain amount of DNA, and like a short amount of those is used to transcribe, then that can be also done quickly. So you can really make all of these work if you're reasoning through them and thinking about the time frame that you want to focus on. Okay, and then last but not least, we're going to talk about the cell cycle and what controls it specifically. So checkpoints in the normal cell cycle prevent cells from going through division if problems occur. For example, if the DNA is damaged, what forms do the checkpoints take? That is, how do they control whether or not the cell division occurs? So there are many different checkpoints that occur in interphase, which comes before prophase. They're called G1, G2, and M phase checkpoints. And they are controlled by CDKs, which are known as cyclic dependent kinases. And so the ratio of the CDKs to their cyclins determines the activity. The activities of the different CDKs control the cell cycle and can move past a given stage of the cell cycle only when the appropriate checkpoint's CDK has been activated. And I will draw this out. Alrighty. And moving on, so in the space below, develop a handout diagram to explain these checkpoints and work under normal conditions. Your diagram should include a description of each checkpoint, where it acts in the cell cycle, and what each controls during cell division. So I have this diagram here just to remember the levels of MPF and cyclin, and that MPF is the mitosis promoting factor, and that we're seeing that in the M phase, or the mitosis phase, that factor is at its highest concentration, and we're seeing cyclin slowly increase until it's used in the M phase and starts going again. So if we were to draw this in a circular fashion, like so, M is really short, there's a checkpoint here, here, here. So each of these checkpoints, G1 has a CDK all the way to G2 that has a CDK. That moves to MPF. That pretty much regenerates CDK because cyclin is degraded. Cyclin comes in here. So these different phases, G1, the cell grows physically and increases the volume of the protein. MPF is the mitosis promoting factor. So it's just a factor that drives mitosis. Yeah, of course. Ask any questions. So it's really about cell growth and size. Size increases. And then G2, or in S phase, sorry, S phase is next. The cell copies the DNA and produces the two sister chromatids and replicates nucleosomes. This is where it copies its DNA. And then finally, G2 is further cell growth. And G2 is emphasized a lot just because that's where the cell goes through its last thing before mitosis, and so that's where it really checks, like, is the DNA replicated properly? Is the cell of normal size? You don't want to prematurely replicate a cell if it's not ready for it yet, just like, people don't induce labor if the baby is not mature enough to be produced. You don't put a cell through replication until it's also mature enough. So that's what these checkpoints do. It's making sure at each point that these CDKs are being produced, and through that is being told signals, hey, let's go, let's go, let's go. So this moves us on to our next question, which talks about cancer, and it says, so cyclin and C decays. Oh, sorry, let's go back. Yes. Cyclin and CDKs work together to drive a cell into cycle transition. And so what MPF is to draw a little thing. If this box right here is the CDK, and this thing up here is the cyclin. This whole complex is MPF. So that's exactly right. I just wanted to draw that for you. So that's why they're called cyclin-dependent kinases. So this thing is dependent on cyclin. One cyclin binds and produces that promoting factor. Then it goes into M1 phase. Cyclin is then degraded. CDK is this is where cyclin's degraded. CDK is reformed, and then this process can go again. Yes, the MPF and mitosis go together. I didn't really draw this well. Honestly, the key when it comes out in this section has a really, really great drawing that I kind of tried to emulate. I didn't do it as well as, but it has this circular diagram, but it's just a bit more organized. So that should help you a ton in this section. I'm sorry about my horrible drawing. Usually I try to do better at those things. And then, so finally, cancer cell results from uncontrolled cell division, and explain how mutations in one or more of the checkpoints might lead into cancer. So remember that these checkpoints are just to make sure the cell is functioning normally and that it's a regular size and should continue through cell division. So if the checkpoints break down, they could allow any cell to continue through the cell cycle continuously. So any cell, any size, anything can divide continuously if the checkpoints break down. And so cancer is basically uncontrolled cell division. So cancer results basically from the breakdown and operation of these checkpoints. And that's also a hint as why we see it more so in geriatric populations or geriatric populations. And thank you very much. Okay. So we're coming to the end. I really appreciate you guys for sticking with me and helping me navigate this term through some of the technical difficulties and helping me get started. Just so you folks know, I will be returning next quarter. We'll be hosting these live streams similarly. Again, if you're continuing through next, if you're continuing through the series, I'll be here going over that material. And of course, thank you so much. I really, really appreciate you guys. If there's any questions, I'll stick around for a minute or two. But if not, really good luck on all of your final exams. I hope you guys enjoy your well-deserved break and that this term has been good for you. And so Twitch sometimes like deletes after a certain amount. Thank you. See you next term. So you can actually go to the Learning Center's YouTube page. And that is where all of my videos will be. You can also watch them on YouTube. So let me get that link for you. I'm just picking it up. Oh. So yeah, okay, that is actually not in my control. So what I do is after these videos, I upload them, and then the Learning Center handles that. So I'm just going to double check and make sure that they upload it today. And I'll ask them to make sure that it's unhidden on the YouTube channel. So thank you for bringing that to my attention. I'll totally email them today. Okay, awesome. If there are no more questions, I'm going to end the stream. And oh, there's no need to apologize. I really do appreciate it. It's no big deal. It's just an email. Yeah, always let me know about stuff like that. I'm totally willing to help. Alright, awesome. Thank you guys. See you next term. Enjoy break.